Another function of the cell membrane is to establish and maintain an electrochemical gradient across the plasma membrane, and we call that the resting membrane potential. This is vitally important for normal muscle contraction and for firing nerve impulses. So cells have an electrical charge difference at the cell membrane. This charge difference represents potential energy, so we call it membrane potential. The membrane potential when a cell is at rest, not doing its job, is called resting membrane potential. There are two conditions that are important in establishing and maintaining that resting membrane potential. So, number one, the cell has an unequal distribution of ions and charged molecules on each side of the membrane. So, the fluid inside the cell, close to the membrane, contains a lot of potassium ions as compared to the fluid just outside the cell membrane. In comparison, the interstitial fluid outside the cell membrane contains a whole lot more sodium than the inside the cell, the intracellular fluid. So more sodium outside, more potassium inside. This is due to those sodium-potassium pumps. Now, the second thing that's important in the resting membrane potential is the amount of positive and negative charges on each side of the membrane. So there's a lot more positively charged ions just outside the cell membrane in the interstitial fluid. Now remember, that's also called the extracellular fluid. And there are more negative charges just inside the cell membrane. That's in the intercellular fluid, or intracellular fluid, excuse me. Now, what that does is to create an unequal distribution of positives and negatives, and also, as we've seen, unequal distribution of the potassium and sodium, and this together establishes an electrochemical gradient. The two most important ions in establishing and maintaining the resting membrane potential are sodium and potassium. And the way that they move across the membrane depends on that electrochemical gradient. So potassium is the most important determinant in the specific value of that resting membrane potential, which can be measured using a device called an electrode, placing electrodes, I should say, from a voltmeter, one outside the cell membrane and one inside the cell membrane. That will allow us to get a relative value to associate, to give us a value to associate with that resting membrane potential. Now, we're not going to have to do any of that. I was just explaining what that means. So the membrane is more permeable to potassium than sodium. So sodium's going to move out of the cell and into the interstitial fluid. Sodium moves into the cells from the interstitial fluid, and that causes the inside of the cell to become more positive. Now, the sodium-potassium pumps are going to be important in maintaining that resting membrane potential that is established by the moving back and forth of the sodium-potassium ions. Now, we're going to get deeper into that later. Let's just get the big picture for now. Now, hopefully you've realized we're still talking about the functions of the cell membrane. So I wanted to pop back to this slide just to remind you of that. We've talked already about how the cell membrane is selectively permeable and provides a barrier between the extracellular fluid and the intracellular fluid. And that boundary, of course, is a physical barrier. We've just finished talking about the electrochemical gradients, and on our next slide, we'll talk about the last function that we'll discuss of the cell membrane, and that is cell-to-cell communication. Remember when we were talking about the proteins in the cell membrane, and some of those were glycoproteins, which means a protein with a sugar attached? Those that are embedded in the cell membrane help cells to be able to communicate between each other, both directly and in response to external molecules. So the direct contact between cells is when one cell actually physically contacts another cell. So this is important in the normal functioning of cells, especially those in the immune system, because the immune system's job is to find things that shouldn't be there and get rid of it. So each of us have a particular type of proteins and particular types of sugars, those glycoproteins, and that's determined by our genes that we get from mom and dad. So because all of our DNA is different, we all make different glycoproteins. So that glycocalyx that is formed from those sugary proteins on the outside of the cell is going to be different in everybody except for identical twins who have the same DNA. Okay? Now, with that said, your immune system floats around, your immune system cells float around through the blood, get out into your body tissues, and they're looking for invaders. When they find those invaders, they sort of survey. Okay, let's look at this glycocalyx. Oh, I don't recognize that. Let's kill it. Or, hey, this is okay. It's just one of her cells. It's supposed to be here. But what if that cell has mutated due to cancer or something has happened? Well, then that cell is very likely to be different and the immune system will also destroy it. Another example of direct cell contact is between a sperm and an egg cell. That's called an oocyte. So during the process of fertilization, the sperm cell recognizes and then binds to the oocyte, and it recognizes it because of that glycocalyx. And then the third example is the direct response or the direct contact that is part of development and growth after we get an injury. So say you cut your hand and you cut just the upper layer of the skin, which is called the epidermis. Well, those cells are going to begin to divide, or actually you could cut even deeper than the epidermis, but the cells in the epidermis are going to start to divide and create new cells. And this is going to continue until that gap that was created by the injury is closed off. So when the damaged tissue has been replaced, overgrowth of skin is prevented because that cell-to-cell contact then inhibits further growth and creation of more cells. Now, other than what we've just talked about, most communication between cells is through ligands. Remember, we talked about this previously. Ligands are molecules that bind to big macromolecule receptors embedded in cell membranes. So ligands that are involved in communication between cells include neurotransmitters that are released from nerve cells and hormones that are released by glands and structures within the endocrine system. So the cell receives information, or the one that does receive the information has the receptor. That receptor binds to the ligand, and that binding initiates mechanisms within the cell for controlling things like growth, reproduction, and other cellular processes. The three types of ligand receptor signaling include channel-linked receptors. So channel-linked receptors are sometimes called chemically gated channels, which simply means that when the ligand binds the receptor, it opens a gate. That gate then will allow something to enter or leave the cell. When a ligand binds an enzymatic receptor, it alters something inside the cell by either activating or turning off an enzymatic pathway inside the cell. And the third type are the G protein coupled receptors. So the G proteins just sort of serve as an intermediate between another action or another molecule. So when the ligand binds the G protein coupled receptors, it simply activates that G protein within the cell, and that in turn causes something else to get turned on or off. In other words, another action in the cell or stopping an action in the cell.